All right, so today we're talking about the Gram stain. All right, the Gram stain is a very popular stain utilized in microbiology in order to identify, or not, well, yes, identify unknowns, but also to better visualize the structure of bacteria. All right. Really briefly, bacteria are very small. They're the most abundant organisms on the earth. Okay, so some general characteristics of bacteria that you should be familiar with is that they are prokaryotic organisms. Okay, so you know that in general we can categorize all living things at the domain level, well, yeah, as either prokaryotic or eukaryotic. There's another domain, archaea, they're very similar to bacteria. Okay, but you don't hear a whole lot about them, but they have some unique characteristics that they kind of needed their own category. All right, but these bacteria here that we'll talk about are prokaryotic organisms. These are unicellular organisms, right? By definition, prokaryotes, they're very simple cells. They don't have a nucleus and all of the other complex membrane-bound organelles, okay? They have a single circular chromosome, okay? They're reproduced by binary fission, okay? These are things that you should have learned about in your Biology 103 course, which is the prerequisite for this course. Binary fission is a form of asexual reproduction, so it only requires one parent. You should definitely, take notes of this information. I do expect you to know these basic general characteristics of bacteria. What is binary fission? Binary fission is basically when a parent cell goes through sort of mitotic division processes to duplicate its DNA and divide to form two new daughter cells. Okay, so this is a form of asexual reproduction. Okay, not to be confused with sexual reproduction. And so structurally, there are two different types of bacteria based on the structure of the bacterial cell wall. Okay, so the bacterial cell wall is a unique structure that not all cells have. Okay, so we have that outer barrier or layer around all cells that we'll call the plasma membrane or the cell membrane, right? All cells have a cell membrane, but not all cells have the cell wall that's going to be a barrier outside of that plasma membrane. Well, bacteria cells have it, and some other organisms have it, like fungi and plants. Okay, so this cell wall in bacteria can vary in terms of the thickness of it. Okay, so we use the Gram stain to help us to determine, what is the bacterial structure in this cell wall. Is this peptidoglycan going to be thick or thin? Okay, that is one of the factors that we look for with the Gram stain. Okay, the outcome of a Gram stain tells us a lot about this bacteria cell. Okay, and we can determine if it's Gram negative or Gram positive based on the color that it ends up being at the end of this stain process. Okay, so let's look at the, and so this cell wall, here's what I want you to know. The cell wall is made up of a peptidoglycan, glycoprotein structure. Gram-negative bacteria are going to have a thin peptidoglycan layer. Okay. Gram-positive bacteria are going to be characterized by a very thick peptidoglycan layer. Okay, you need to write this word peptidoglycan down. Peptidoglycan. Give you a moment to write that down. That's a very important term in microbiology. All right, so this peptidoglycan layer, again, is the major component of the bacteria cell wall. And when we gram stain, okay, when we do the gram stain technique, it's going to be based on the structure of this bacteria cell wall that's made up of this peptidoglycan. It's either going to be a thin layer of peptidoglycan or there's a thick layer, all right? And the color of that gram stain at the end of that process tells us about this bacteria structurally, okay? So the gram stain technique, okay, this name is derived from the bacteriologist who developed this technique, Christian Graham. Okay, the gram stain technique by definition is just a method of staining that's used to differentiate different bacterial species. Okay, so if you went into a lab and you had an unknown culture of bacteria, one of the first things that we can do to try to identify what's in this culture is gram stain it. Okay, it's a very quick process. It takes about 5 minutes. Okay, and it's an important tool for, you know, initial identification and classification of bacteria. So write that down. Why is this such a useful tool? This staining technique allows us to differentiate between different species of bacteria and helps us to begin identifying and classifying bacteria. Okay. Now, again, you hear me using the term to begin classifying. It's not an end all be all. We can classify it generally as gram positive, gram negative based off of this gram stain technique, but we will use additional techniques to further, you know, identify things and give it an actual, to be able to designate actual nomenclature to it, genus and species. We'll look for some other things, but we can start out with this gram stain technique. Maybe we can look for some other characteristics to help us maybe identify it, looking at morphology, arrangement under the microscope, you know, motility. There are a number of other characteristics that we can look at to help us identify it, but we typically start with the gram stain. Okay, we talked about the microscope last week. This gram stain technique, this procedure is based on the ability of the microorganism to retain the colors of the dye that's used during this reaction. Okay, so in essence, we're going to have our bacteria fixed to a microscope slide, and we're going to add some series of dye drops to it, do a few washes. Again, it's a very quick process, takes about 5 minutes. You're adding various dyes, and we'll talk about it in just a second. And then we just go and look at it under the microscope. Okay, and again, we're looking for some, you know, important morphological features and arrangements, and these stains help us to see these. Okay, but depending on the color that these cells retain at the end of this procedure will help us to identify it. And at the end of the procedure, we can determine if the cells are gram positive or gram negative, and that tells us some initial identification features. Okay, and so the gram stain technique then is used to differentiate bacteria into two large groups. Okay, gram positive or gram negative. Okay, that's what we get from this technique. We can identify or categorize bacteria into two large groups. Okay, let's look at how this procedure goes. The things you'll need to do a gram stain procedure, you should write these things down. You're going to need a culture or a specimen. Okay, that's going to be your bacteria. Okay, this is your general necessities, and we'll get into a little bit more detail, but in general, okay, formulate your mind. If we were in the lab, we would do this. You would need your bacteria, right, your culture. You would need your gram staining reagents, and we'll talk about those reagents in just a moment. There are 4. You'll need a microscope slide, right? You need a heat source, and we'll talk about the purpose of that heat source in just a second, which is basically to fix the bacteria on the slide so that it doesn't wash off as you're, you know, adding your dyes and doing your rinses and so forth. You don't want to lose your culture off your slide. And then you'll use the inoculating loop or a swab, and then you need a microscope, you know, once you're done to visualize it. Okay, so write those things down. You need your bacteria, you need your staining dyes, okay, your gram stain reagents, you need a microscope slide, a heat source, and the microscope. And I keep getting a message that my internet is kind of going in and out. This is being recorded and I will post it when we're done. So just bear with me. So, gram staining reagents. In your notes, you need to write down these 4 gram stain reagents. You cannot do a gram stain procedure without these 4 reagents. Crystal violet. Write down in your notes these 4 reagents. Crystal violet. And next to it, write down that it's the primary stain. This name is important. Crystal violet is a primary stain. This is going to be the first stain that we start with, the first dye that we use to saturate our cells. Crystal violet is the primary stain. Iodine. This is the next reagent we need for gram stain. Iodine is a mordant. Write the word mordant next to iodine. Mordant is spelled M-O-R-D-A-N-T. D as in dog, mordant. Does anyone know what is the function of a mordant? Okay, the function of the mordant is to help to fix this primary stain into the cells. Okay. The next reagent, ethanol, alcohol. It's a decolorizer. Write that next to alcohol. It's a decolorizer. Okay, it's a decolorizer. Okay, this is the step where we're attempting to remove that primary stain from our cells. Okay, alcohol is a primary, the decolorizer. And then last, safranin. Safranin is the last gram stain reagent that you need here. It's our secondary stain. So after the decolorizer step, we add a second stain to see if the cells will retain this stain or not, okay? And this is going to ultimately determine if these cells end up one of 2 colors. I have a visual. Yes.
You're breaking up. I don't know if it's just me.
Yes, I just made a statement about that. I'm seeing it here. I'm not sure why my internet is not stable, but this is being recorded and I will upload it for you. All right, let's look at what happens during a gram stain procedure. So, yes, sir.
Won't the recording also be glitching as well?
I don't think so, because it's recording onto the computer. Okay, let's just try and keep going. I don't know what else I can do right now. I don't have any other way to adjust my internet connection. Gram staining. All right, so let's look and see what happens. So we start out with these bacteria cells, okay? Generally speaking, they're gonna appear sort of a clear translucent color if you just attempted to look at them under the microscope, okay? We're gonna go through our fixation process. Write down this term, fixation. Fixation is when you are adhering your bacteria cells to your slide, and you do need the heat source for that purpose, okay? So we can use a Bunsen burner or so to help fix our cells onto the slide so that they don't rinse off. So after fixation happens, okay, so we have our microscope slide, we've added our bacteria, and then we fixed our bacteria onto the slide. We're gonna start the gram stain procedure, all right? The first thing that we're gonna do is add the primary stain, which is crystal violet. So you're going to add a few drops of crystal violet onto your microscope slide where your cells are. Okay, jot that down. Then you're going to add a few drops of iodine. Okay, well, you rinse, and then you add a few drops of iodine. Okay, this iodine, as I mentioned before, it functions as a mordant, and it helps to fix that dye into the cells. Okay, so you're going to add your iodine. All right. Next, we're going to use some alcohol. You're going to have a few drops. After we rinse off our iodine, we're going to add a few drops of alcohol to the slide. Okay, this is our decolorizing agent. Okay, decolorizer is basically going to attempt to remove that primary stain. And depending on if these are gram-positive or gram-negative bacteria, the colors of these cells would change after this step. Okay, if it's a gram-positive bacteria, it's going to retain that primary stain, so it's going to stay purple. Okay, after the decolorizing step though. But however, if it's a gram-negative bacteria, when we add the decolorizer, the gram-negative cells will not retain that primary stain. Okay, and they're going to start to appear a clear color again because we've rinsed off the primary stain. And then we're going to add that counter stain or secondary stain, which is safranin. Okay, safranin, again, at this point, if we have gram-positive bacteria, the cells will not take up the safranin, and they're going to stay purple. Okay, if they're gram-positive. But if they're gram-negative, then these cells will appear pink after adding that counter stain. Okay, so safranin is sort of a we say a counter stain. If the primary stain is removed, which is crystal violet, after the decolorizer, and then we add safranin, so the cells are going to end up pink instead of purple. Okay, and this is how we would make our final determination of the cells that are present, in that culture. All right, the cells are going to either end up purple, which means that they're gram-positive. Okay, or they're going to appear sort of a pinkish red when you look under the microscope, and that's going to indicate that they're gram-negative. And this is just a visual here to show you what that process looks like. Again, it's a very simple process where we're adding, just some drops of dye, a few drops of iodine. We rinse it, use our decolorizer, and we, finally, look at those cells under the microscope after we've, rinsed off all of our dyes and sort of blotted dry. And we look at it under the microscope. Okay, so real quick, I know this connection is not the best, but when you're looking under the microscope, gram-positive bacteria are going to stain purple. Okay, right here is a microscope image of what some gram-positive, bacteria will look like under the microscope. Okay, and so they are looking purple because they retained that primary dye. Make sure you understand that concept, that when we went through the entire procedure and we tried to decolorize, the crystal violet was retained, or it stayed on those cells. Okay, they will not decolorize when they are washed with alcohol. Okay, some examples of gram-positive bacteria, you need to jot these down in your notes, are Bacillus, Streptococcus, Staphylococcus. These are all general examples of gram-positive species. In contrast, gram-negative bacteria are going to stain sort of a pink or a red color. Okay, that's due to the retention of that counter stain, safranin. Okay, so this is a microscope image of what a gram-negative bacteria would look like under the microscope. It's going to appear sort of a pinkish red. Okay, because what happened is that these cells did in fact decolorize after washing with alcohol. Okay, examples of gram-negative is E. coli. You probably hear about that a lot. Escherichia coli, Pseudomonas aeruginosa. These are examples of gram negative organisms. Okay, and so this was just, again, an opportunity for you to learn about one of the very basic stains. Okay, there are a few review questions that I want you to do at the end. All right, and then I'm also getting ready to post a exercise for you in the Blackboard course. Okay.